This review paper with title Recent Advances in Site-Specific Lipid Nanoparticles for mRNA Delivery highlights how the success of mRNA vaccines has spurred the development of innovative therapies, focusing on the use of LNPs to protect and deliver mRNA effectively to targeted cells. By enhancing the precision of mRNA therapy, these site-specific LNPs not only improve therapeutic outcomes but also minimize unwanted side effects. Dive in to explore the latest strategies and future prospects in this fascinating field. Here are 3 example questions you can ask. 1. What are the main advantages of using site-specific lipid nanoparticles for mRNA delivery? 2. How do local and systemic administration methods differ in their effectiveness for mRNA therapy? 3. What future developments are anticipated in the field of mRNA therapy and lipid nanoparticles? Here are the key and brief points for the abstract section. The success of mRNA vaccines during the COVID-19 pandemic has accelerated the development of mRNA therapy. mRNA serves as a template for protein synthesis but is unstable and requires suitable carriers for effective in vivo delivery. LNPs are utilized to protect mRNA from degradation and enhance its intracellular delivery. Recent advancements focus on developing site-specific LNPs that can accumulate in targeted organs, tissues, or cells through local or systemic administration. These site-specific LNPs improve the efficiency of mRNA therapy and reduce off-target adverse effects. The review summarizes recent strategies for site-specific mRNA delivery and discusses the prospects for future mRNA therapy developments. Here are the key and brief points for Figure 1 based on the provided document. Composition of mRNA-loaded LNP. The figure illustrates the components of lipid nanoparticles LNPs used for mRNA delivery, which include ionizable lipid, facilitates the encapsulation and delivery mRNA. Helper lipid, supports the stability and functionality of the LNP. Cholesterol, enhances the structural integrity and fluidity of the nanoparticle. PEG lipid, provides a protective layer to improve circulation time and reduce immunogenicity. Administration routes. The figure highlights various routes of administration for LNPs, including local administration, direct injection into specific tissues or organs for targeted therapy. Systemic administration, intravenous injection allowing for broader distribution throughout the body. Targeted delivery. The figure emphasizes the goal of achieving organ, tissue, or cell-specific mRNA delivery, which enhances therapeutic efficacy and minimizes systemic side effects. Therapeutic effects. The illustration suggests that effective delivery of mRNA via LNPs can lead to localized or systemic therapeutic effects, depending on the administration route. Summary of Table 1 findings. Overview of site-specific LNPs, presents strategies and advancements in developing site-specific lipid nanoparticles LNPs for mRNA delivery. Targeting mechanisms, O-ligand-receptor interactions, use of specific ligands for receptor binding on target cells. O-structural modifications, altering lipid composition or structure to enhance targeting. Administration routes, effectiveness of local versus systemic delivery for targeting specific organs or tissues. Therapeutic application, O-cancer therapy, targeting tumor cells for localized treatment. O-bone tissue engineering, delivering mRNA to bone tissues for regeneration. Performance metrics, includes transfection efficiency, mRNA expression levels, and reduction of target effects. Future directions, ongoing research aimed at optimizing LNPs for more precise and effective mRNA delivery. This summary highlights the strategies, applications, and performance metrics of site-specific LNPs in mRNA therapy as detailed in Table 1. Highlights for Figure 2 include, enhanced mRNA delivery. the figure demonstrates that the design of lipid nanoparticles LNPs significantly improves the delivery efficiency of mRNA to target cells. Targeting efficiency, results indicate that LNPs functionalized with specific targeting ligands show increased uptake by target cells compared to non-targeted LNPs. Cell-specific expression, the figure illustrates that mRNA delivered via targeted LNPs leads to higher levels of protein expression in specific cells, confirming the effectiveness of the targeting strategy. Reduced target effects, the results suggest that site-specific delivery minimizes the expression of mRNA in non-target cells, thereby reducing potential side effects associated with systemic delivery. Comparison of formulations, the figure may include comparisons between different LNP formations, highlighting which compositions yield the best performance in terms of mRNA delivery and expression levels. Therapeutic efficacy, the results indicate that targeted LNPs can achieve desired therapeutic effects in specific tissues or organs, supporting their potential for clinical applications. These results summarize the key findings illustrated in Figure 2, focusing on the effectiveness of LNPs in mRNA delivery and their targeting capabilities. Here are the highlighted points for selective organ targeting SORT nanoparticles and their organ-specific targeting delivery after intravenous injection based on Figure 3, Introduction of SORT nanoparticles. SORT nanoparticles are designed to achieve selective delivery of mRNA to specific organs after intravenous administration. Composition of SORT nanoparticles, the formulation includes a fifth component, SORT lipids such as DOTAP, 18PA, and DOTAP, which are crucial for enhancing organ-specific targeting. Mechanism of action, the targeting mechanism relies on the interaction between the SORT lipids and specific receptors or proteins present in the target organs, facilitating the uptake of nanoparticles. Biodistribution, the figure illustrates how varying the proportion of cationic, anionic, and ionizable lipids in the formulation affects the biodistribution of LNPs to different organs, such as the lung, spleen, and liver. Optimized formulations, results indicate that specific ratios of lipids, e.g. 50% cationic SORT lipid for lung targeting, 30% anionic SORT lipid for spleen targeting, and 20% ionizable cationic SORT lipid for liver targeting enhance the delivery efficiency to the intended organs. Clinical implications, the ability to achieve selective organ targeting through SORT nanoparticles has significant implications for improving the therapeutic efficacy of mRNA-based treatments while minimizing off-target effects. These points summarize the key aspects of SORT nanoparticles and their effectiveness in achieving organ-specific delivery after intravenous injection, as depicted in Figure 3. Summary of Figure 4 findings. Bisphosphonate lipid BP, core component designed for binding calcium ions in bone, crucial for bone targeting. Calcium ion chelation, BP head group has strong affinity for calcium in bone's hydroxyapatite, aiding localization and retention. LNP composition, BP lipid, facilitates bone targeting. Helper lipid dope, supports LNP formation and stability. Cholesterol, ensures structural integrity and fluidity. PEG lipid, prolongs circulation time and reduces immune detection. Systemic administration, designed for intravenous injection to circulate and accumulate in bone due to BP calcium interactions. Targeted delivery, aims for delivering mRNA or nucleic acids to bone cells, beneficial skeletal diseases. Therapeutic applications, intended for mRNA delivery to promote bone regeneration or treat bone disorders. This design leverages bisphosphonate's unique properties to enhance the specificity and efficacy of mRNA delivery to the bone microenvironment. The preparation of leukocyte-specific lipid nanoparticles LNPs, as depicted in Figure 5, involves several key steps aimed at enhancing the targeting of LNPs to leukocytes' white blood cells. Here is a brief overview. Initial LNP formation. The process begins with microfluidic mixing of lipids and therapeutic mRNA to create unmodified LNPs. This step ensures the encapsulation of mRNA within the lipid matrix, forming stable nanoparticles suitable for delivery. Incorporation of targeting ligands. To achieve leukocyte specificity, a modular targeting platform known as ecent antibody-specific targeting micelle is introduced. This platform allows for the conjugation of specific antibodies or ligands to the surface of the LNP's conjugation process. The ecent micelle is mixed with the unmodified LNPs, facilitating the attachment of leukocyte-specific ligands such as antibodies that bind to receptors on leukocytes. This modification is crucial for directing the LNPs to leukocytes upon systemic administration. Targeting mechanism. The presence of these specific ligands on the surface of the LNP enhances their uptake by leukocytes through receptor-mediated endocytosis, allowing for more efficient delivery of the encapsulated mRNA to these immune cells. Therapeutic potential. The leukocyte-specific LNPs are designed for applications in immunotherapy, vaccine delivery, or treatment diseases involving the immune system, where targeted delivery to leukocytes can enhance therapeutic efficacy and minimize off-target effects. Overall, the preparation of leukocyte-specific LNPs represents a sophisticated approach to improve the precision of mRNA delivery to immune cells, leveraging targeted ligands to achieve specific cellular uptake and therapeutic outcomes. Summary of findings for Figure 6. Background on FAP and cardiac injury. Fibroblast activation protein FAP is overexpressed in activated cardiac fibroblasts, indicating cardiac injury and fibrosis, making it a therapeutic target. CAR T-cell construction. The therapy involves creating CAR T-cells that recognize and target FAP-expressing cells by delivering CAR mRNA specific to FAP into T-cells. Anti-CD5 antibody-modified LNPs. LNPs are modified with anti-CD5 antibodies to target T-cells, allowing specific binding and uptake of CAR mRNA by T-cells. Tragocytosis mechanism. CAR T-cells express the CAR on their surface, enabling them to target and eliminate FAP-expressing cells through tragocytosis, addressing cardiac injury. Improvement of cardiac function. Targeting activated cardiac fibroblasts reduces fibrosis and remodeling, potentially reversing cardiac injury and improving heart function. Therapeutic implications. This strategy combines CAR T-cell technology with targeted delivery systems, offering a promising treatment for cardiac injuries related to fibroblast activation. This innovative approach leverages the specificity of CAR T-cells and the targeted delivery capabilities of modified LNPs to address cardiac injury effectively. Conclusion. Targeting FAP. The overexpression of fibroblast activation protein FAP in activated cardiac fibroblasts serves as a significant marker for cardiac injury, making it a viable target for therapeutic strategies. CAR T-cell construction. The development of CAR T-cells that specifically recognize FAP is facilitated through the use of lipid nanoparticles LNPs for the delivery of mRNA encoding the CAR, enhancing the precision of the therapy. Enhanced delivery with anti-CD5 modification. The modification of LNPs with anti-CD5 antibodies allows for targeted delivery to T-cells, improving the uptake and expression of the CAR. Mechanism of action. The ability of CAR T-cells to tragocytose FAP from activated cardiac fibroblasts enables effective targeting and elimination of these cells, addressing the underlying causes of cardiac injury. Therapeutic implications. This approach holds promise for reducing cardiac fibrosis and improving cardiac function, representing a significant advancement in the treatment of cardiac injuries. Perspectives. Further research, continued investigation into the long-term effects and safety of CAR T-cell therapies targeting FAP is essential to establish their clinical viability. Broader applications. the methodology could be adapted for targeting other markers associated with various diseases, expanding the potential of CAR T-cell therapies beyond cardiac applications. Combination therapies, exploring the combination of CAR T-cell therapy with other treatment modalities, such as immunotherapies or traditional pharmacological approaches, may enhance therapeutic outcomes. Personalized medicine, the development of personalized CAR T-cell therapies based on individual patient profiles could lead to more effective and tailored treatments for cardiac and other diseases. Regulatory considerations, addressing regulatory challenges and ensuring compliance with safety standards will be crucial for the successful transition of this technology into clinical practice. In conclusion, recent advances in site-specific lipid nanoparticles for mRNA delivery, published in ACS Nanoscience 2023, showcases the significant strides made in the field of mRNA therapy. The advancements in lipid nanoparticle LNP technology highlight the potential for targeted and efficient delivery of mRNA, paving the way for innovative treatments for a variety of diseases. These breakthroughs not only enhance the therapeutic efficacy but also reduce potential side effects, marking a new era in precision medicine. We encourage you to dive into the full paper to explore these exciting developments in detail. Don't miss out on the opportunity to stay informed about the future of mRNA delivery technologies. If you found this paper insightful, please like and subscribe to our channel for more informative videos. Share this video with your friends and colleagues, and join the conversation by responding to the featured Iranian music in the comments. Your support helps us bring more cutting-edge content to the scientific community and beyond.